Cell division. The continuity of life from one cell to another has its foundation in the reproduction of cells by way of the cell cycle. The cell cycle is an orderly sequence of events that describes the stages of a cell's life from the division of a single parent cell to the production of two new daughter cells. The mechanisms involved in the cell cycle are highly regulated. Genomic DNA. Before discussing the steps a cell must undertake to replicate, a deeper understanding of the structure and function of a cell's genetic information is necessary. A cell's DNA, packaged as a double-stranded DNA molecule, is called its genome. In prokaryotes, the genome is composed of a single, double-stranded DNA molecule in the form of a loop or circle. Figure 1. The region in the cell containing this genetic material is called a nucleoid. Some prokaryotes also have smaller loops of DNA called plasmids that are not essential for normal growth. Bacteria can exchange these plasmids with other bacteria, sometimes receiving beneficial new genes that the recipient can add to their chromosomal DNA. Antibiotic resistance is one trait that often spreads through a bacterial colony through plasmid exchange. In eukaryotes, the genome consists of several double-stranded linear DNA molecules. Figure 2. Each species of eukaryotes has a characteristic number of chromosomes in the nuclei of its cells. Human body cells have 46 chromosomes, while human gametes, sperm or eggs, have 23 chromosomes each. A typical body cell, or somatic cell, contains two matched sets of chromosomes, a configuration known as diploid. The letter N is used to represent a single set of chromosomes. Therefore, a diploid organism is designated 2N. Human cells that contain one set of chromosomes are called gametes, or sex cells. These are eggs and sperm, and are designated 1N, or haploid. Matched pairs of chromosomes in a diploid organism are called homologous. Same knowledge. Chromosomes. Homologous chromosomes are the same length and have specific nucleotide segments called genes in exactly the same location, or locus. Genes, the functional units of chromosomes, determine specific characteristics by coding for specific proteins. Traits are the variations of those characteristics. For example, hair color is a characteristic with traits that are blonde, brown, or black. Each copy of a homologous pair of chromosomes originates from a different parent. Therefore, the genes themselves are not identical. The variation of individuals within a species is due to the specific combination of the genes inherited from both parents. Even a slightly altered sequence of nucleotides within a gene can result in an alternative trait. For example, there are 3 possible gene sequences on the human chromosome that code for blood type: sequence A, sequence B, and sequence O. Because all diploid human cells have 2 copies of the chromosome that determines blood type. The blood type, the trait, is determined by which 2 versions of the marker gene are inherited. It is possible to have 2 copies of the same gene sequence on both homologous chromosomes, with one on each. For example, AA, BB, or OO, or 2 different sequences, such as AB. Minor variations of traits, such as blood type, eye color, and handedness, contribute to the natural variation found within a species. However, if the entire DNA sequence from any pair of human homologous chromosomes is compared, the difference is less than 1%. The sex chromosomes, X and Y, are the single exception to the rule of homologous chromosome uniformity. Other than a small amount of homology that is necessary to accurately produce gametes, the genes found on the X and Y chromosomes are different. Eukaryotic chromosomal structure and compaction. If the DNA from all 46 chromosomes in a human cell nucleus was laid out end to end, It would measure approximately 2 meters. However, its diameter would be only 2 nanometers. Considering that the size of a typical human cell is about 10 micrometers. 100,000 cells lined up to equal 1 meter. DNA must be tightly packaged to fit in the cell's nucleus. At the same time, it must also be readily accessible for the genes to be expressed. During some stages of the cell cycle, the long strands of DNA are condensed into compact chromosomes. There are a number of ways that chromosomes are compacted. In the first level of compaction, short stretches of the DNA double helix wrap around a core of 8 histone proteins at regular intervals along the entire length of the chromosome. Figure 3. The DNA histone complex is called chromatin. The bead-like histone DNA complex is called a nucleosome. And DNA connecting the nucleosomes is called linker DNA. A DNA molecule in this form is about 7 times shorter than the double helix without the histones. And the beads are about 10 nanometers in diameter. In contrast with the 2 nm diameter of a DNA double helix. The next level of compaction occurs as the nucleosomes and the linker DNA between them are coiled into a 30 nm chromatin fiber. This coiling further shortens the chromosome so that it is now about 50 times shorter than the extended form. In the third level of packing, a variety of fibrous proteins is used to pack the chromatin. These fibrous proteins also ensure that each chromosome in a non-dividing cell occupies a particular area of the nucleus that does not overlap with that of any other chromosome. See the top image in Figure 2. DNA replicates in the S phase of interphase. After replication, the chromosomes are composed of 2 linked sister chromatids. When fully compact, the pairs of identically packed chromosomes are bound to each other by cohesin proteins. The connection between the sister chromatids is closest in a region called the centromere. The conjoined sister chromatids, with a diameter of about 1 micrometer, are visible under a light microscope. The centromeric region is highly condensed and thus will appear as a constricted area. Section summary. Prokaryotes have a single circular chromosome composed of double-stranded DNA, whereas eukaryotes have multiple linear chromosomes composed of chromatin surrounded by a nuclear membrane. The 46 chromosomes of human somatic cells are composed of 22 pairs of autosomes, matched pairs, and a pair of sex chromosomes, which may or may not be matched. This is the 2N or diploid state. Human gametes have 23 chromosomes or one complete set of chromosomes. A set of chromosomes is complete with either one of the sex chromosomes. This is the N or haploid state. Genes are segments of DNA that code for a specific protein. An organism's traits are determined by the genes inherited from each parent. Duplicated chromosomes are composed of 2 sister chromatids. Chromosomes are compacted using a variety of mechanisms during certain stages of the cell cycle. Several classes of protein are involved in the organization and packing of the chromosomal DNA into a highly condensed structure. The condensing complex compacts chromosomes, and the resulting condensed structure is necessary for chromosomal segregation during mitosis. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.